smokes. Okay, for those of you who are running late, welcome to the session on small preliminary data sets. Cerebral lateralization is this trait that was thought to once be uniquely human-handedness. It's now thought to be ubiquitous across vertebrates. It's sexually dimorphic and it's mysterious. It's prenatal testosterone exposure, autism, schizophrenia. Some biologists think of this as a left hemifield bias for aggression, predator vigilance, and a right eye bias for foraging and prey capture. Others think of it as like that approach is mediated by the left hemisphere and avoidance by the right hemisphere, or that focused attention is done by the left hemisphere and broad vigilance by the right. So this animal here is being efficient at what it's doing because it's using its dedicated hemisphere for foraging and its other dedicated hemisphere for predator vigilance. There's a lot of individual variation, both in the strength and direction of these cerebral lateralizations. For example, more lateralized chicks detect predators faster. They also forage better under threat of predation. And fish descended from high and low predation pressure populations differ in their strength of this preferred eye use. So the standard fish-handedness task would send a fish down an alley to a transparent barrier, it either turns left or right. And Colin Brown showed that fish descended from high predation pressure populations are more consistent in their choices if they're male, and fishes from low predation populations are more consistent in their choices if they're female. So we do the same sort of thing with aggression. We do an aggression task, about half the fish aggress towards the thing in convict cichlids, and non-aggressive females tend to turn left, as do aggressive males, right? Now, the strength of this effect is much larger when we look at the absolute strength. So that non-aggressive females are more consistent in their choices, they're more strongly eyed, as are more aggressive males. And this is something like what we find in humans, where extremely right-handed individuals are more aggressive than weakly right-handed individuals, who are just like left-handed individuals who are all weakly left-handed. Right? We don't find this effect in females. So in convict cichlids, we found that this trait is linked to aggression, is linked to boldness, and is linked to a response to emotional valence. So brain, this is famously asymmetric epithalamic structure, as you all know. It's evolutionary ancient. It's on this pathway that contains all these inputs from the limbic system, and it puts out to motor, and it's strongly inhibitory. Neuroscientists in the house, raise your hand. Woo! Seeing none, moving on. All right. So we measure the size of the habenula on either side in a cohort of geophagus, and we plot this against how big the fish is. And the bigger the fish, they're sexually dimorphic, males lack, females open. The bigger the male is, the more the left habenula tends to be larger. The smaller the female is, the more the right habenula tends to be larger. And right about where you get the size cut off is symmetry. Eerie. Yeah? Oh, we also find that the magnitude of the asymmetry of the habenula correlates with the magnitude of the absolute value of their handedness. More asymmetrical brains, more asymmetrical behavior. All right, so do differences in growth rate, these are all the same age, they grew up together, influence habenula asymmetry and thus produce variation in personality. So the habenula is basically this nucleus that says, whoa, right? And it's strongly asymmetrical between the hemispheres. So it would make sense if the fastest growing fish were bold, aggressive males with asymmetrical habenula, the less fast growing fish were more cautious males with more symmetrical habenula, and the slower growing fish were females with cautious, and the slowest ones are bold, fast maturing females with asymmetrical habenula. This is a picture of my study species. First off, does growth rate determine sex? Right? And in cichlids, Francis and Barlow had this pretty influential paper showing that size determines sex rather than sex determining size, and Oldfield shows no, this is not the way that it goes. So these, Francis and Barlow's experiment requires that size hierarchies are stable. And we don't find size hierarchies to be really stable in cohorts of convict cichlids, although we tagged them pretty old here, right? We waited until they were big enough to be tagged. So we did this experiment to see if size determines sex at younger ages. We take one cohort of wild types, we divide them up into two lots. We're going to grow up this lot with larger leucistic fish, and this lot with smaller leucistic fish. So we force these ones to the bottom of the size hierarchy, we force those guys to be top of the size hierarchy. And then when we count the number of males and females, when we can sex their gonads by histology, and we find that wild type smaller produces more females, and wild type larger produces more males, which is what you would expect if growth, relative position in the size hierarchy determines sex. Now, it could also be due to differential mortality, right? Because they didn't all make it, but we don't think so. If we look at the size, the same thing works for weight and length. Here's our two experimental groups, right? And this is females, males, statistics they were grown with, females, males, and the males from the group where they were made to be larger turned to be larger, and it doesn't look like this distribution is part of that one with slight death. All right, so handedness in convicts and fry. We changed the handedness task. Cerebral lateralization with this mirror octagon. They're swimming around. They can see their reflection with one eye, and we let them go for 5 minutes, and we measure the amount of time they spend with one eye or the other looking at their reflection. And we find in a cohort of fry at 6 months of age that the bigger the fish is, the more it likes to use its left eye. The smaller the fish is, the more it likes to use its right eye. And we do it again in the second cohort, and we find the same darn thing. The bigger the fish is in this cohort, the more it likes to use its left eye. The smaller it is, the more it likes to use its right eye. So we do it a third time. Well, you know, we can't win them all. But we've got this bruiser here who's messing everybody up, right? They're all stressed out of their gourds. It's foreshadowing. And we find a correlation between the magnitude of the habenula asymmetry and the magnitude of behavior, but because God hates me, it's completely nonsensical connection. That's just a onesie. All right, so are the faster growing fish becoming male? So here we've sexed them, and the open ones are males, the dark ones are females, and the gray ones are indeterminate, and it's not like we're catching them as the males are growing faster, right? Bollocks. So we do it again in the other cohort. Bollocks again. Wow. All right, so maybe if we experimentally manipulate the growth rate of the fry, we will find, holy smokes. How's everyone doing today? I've had lots of coffee. I'm really nervous. I put a second talk on the end of my talk just in case I ran out of time. Who'd have thought? All right. So, for the first 60 days or 30 days of the fry's life, we provide them with a stressor, right? And this stressor influences fear and anxiety behaviors in ways that I wouldn't have thought I would have time to discuss at length, but it influences their behavior at adulthood. Now, we started out using these manipulations where we're exposing them to Shrek stuff, right? Which is this chemical pheromone, this damage-induced chemical release, and it's been shown by others in convict cichlids that exposure to this chemical speeds the maturation, stunts the growth, and speeds maturation. Which sounds like just a perfect manipulation to do these things. And like I said, the talk of small preliminary data sets, we find that the magnitude of the habenula asymmetry correlates with emerge time so that we get more asymmetrical habenula, bolder fish, right? So the shtick, this is our idea, right? If this hemisphere, this nucleus, is allowing one hemisphere to tell the other to shut up, that the more asymmetrical it is, the more you have a brain that can make unilateral decisions. It's almost a pun, I'm not even sure, right? But if the two habenula are symmetrical, then each hemisphere is equal in its ability to tell the other one to shut up, and you have a brain that has to sort of make decisions by consensus. So if you have a unilateral decision-making fish, it should make decisions quickly, but maybe more error-prone, right? Bold, aggressive, proactive stress coping style. Kamsa. Now, one would hope, one would think, life would make so much sense. If you stress out an animal and it's going to mature faster, you're pushing it to a live fast, die young strategy, right? And you should get bolder, quicker emerge times. But we don't find that. Our stressed individuals seem to fall on the same sort of line as our unstressed. Now, it could be that we're not stressing them out enough, but these guys are pretty stressed. I mean, the other fear and anxiety behaviors, open field, elevated plus maze, it's underwater, we call it submerged plus maze, we find it very amusing to call it that, show that there are definite effects of this treatment, but it doesn't seem to be influencing habenula asymmetry. Oh, yes, habenula asymmetry. And we do somewhat soothingly find our usual relationship where the magnitude of the habenula asymmetry correlates with the magnitude of the asymmetry in behavior. Right? And again, on the other hand, we find no effect in our small preliminary data set of the stress treatment on behavioral asymmetry either, which is kind of odd and depressing. Amusing. So, the habenula seems to have a role in this shy, bold, proactive, reactive, stress coping style personality dimension. That seems to be the big personality dimension in non-human animals. We think that growth rate is part of this spectrum, right? That animals within a cohort that mature fast should be bold. We find consistently that the habenular asymmetry correlates with behavioral variation, or at least behavioral asymmetries. Behavioral lateralization correlates with size and convex cichlid fry, but not apparently with habenular lateralization. So if this relationship comes, it comes later, sort of the same way that Oldfield, when he was looking at the development of sex as a function of size, concluded that while you might find evidence later on that looks as if growth rates determine sex, there's some discontinuity in the middle. And I suspect that this discontinuity is the same one that's related to the habenular lateralization and size. The development is not a trajectory that goes straight through. There's some organizing phase in the middle of the midst. In conclusion, the development of habenular asymmetry is unsimple, rather like the development of sex, probably, and fascinating. All right, and with that, I would like to thank NSERC for giving me money, former grad students and collaborators, former students who worked on human stuff, and undergraduates that slaved away in the lab for me. And you for your attention and tolerance. Thank you very much. I'd love to take questions. You all have burning neuroscience questions, and I've cowed you into not asking them by making fun of neuroscientists. Yes?
Thanks. So the stress manipulation presumes that it's plastic responses, that they're not constrained by some underlying genetic constraints. So,
well, I don't know. I think that we find response in HPA axis-like stuff suggests to me there's an organizing effect there, that we stress those suckers out, and it's sort of like growing up in a hybridation environment, right? We're subjecting them to essence of shredded sibling, and it does influence their stress, behavior. It just doesn't seem to influence this stuff, at least not with the sample sizes that we have. Which is sort of totally unlike what one finds with humans, right? Because if that happens, it makes sense. But human personality isn't like that, right? You can't find any sensible variant of upbringing style or anything short of like a super nasty, abusive childhood that influences personality variables in humans. It's just, humans don't make sense. It would be soothing maybe if fish did, but make them maybe less useful as a model. I'm not giving away the talk. Handedness in humans. Strength of handedness in humans? It's weird. It doesn't work that way. All these strength of handedness in humans things, I think they're measuring separate dimensions. You know that snowboard thing? People snowboard one way or the other? Totally uncorrelated with handedness. Same thing with the way they hold a hockey stick. Totally uncorrelated with handedness. Or snowboard. Different. Thank you very much.